About the same time that Punnett was experimenting with sweet peas, American geneticist Thomas Morgan was conducting similar experiments on the fruit fly Drosophila. Morgan led a remarkable team of young scientists. Their work in the fly room, as their lab was called, was some of the best genetics research work of the time. Much of our understanding of crossing over comes from the studies carried out by Morgan and his students. Together, they discovered that body color and wing shape of fruit flies didn't assort independently. Eventually, they proposed that perhaps the genes were carried on the same chromosome. In other words, that the two characteristics were linked.
Have you solved the problem yet?
Well, does it have anything to do with the way genes behave?
You're getting warmer. Maybe these models of chromosomes will help. Remember Sutton and Boveri?
Yeah.
Both of them suggested that somehow genes might be linked. If you look at this chromosome, it has the gene for purple flower, capital P, and also the gene for long pollen, capital L. Its homologue has the small p and the small l genes.
Okay, so that means that the two genes are linked together on the same chromosome, and they would remain linked together during meiosis, right?
Correct. Now, Sandra, how many different gametes can you get from these chromosomes?
Well, on one gamete, we'd have capital P and capital L, and the other gamete would contain the genes small p and small l.
Good. So, now, if two plants that were homozygous mated, what kind of offspring would we expect in the first generation?
Well, they'd all be the same, wouldn't they? Purple long.
Good. Now, Scott, can you review for us the ratios that we would expect if two plants from the F1 generation mated?
Sure. We ended up with a ratio of 9 purple long to 3 purple round to 3 red long to 1 red round. But our results don't match up with Punnett's experimental results.
Right. Let's take another look at this chart. But this time, we'll cover the data in the middle. Now, that will leave us 381 offspring, 296 of which are purple long, and 85 of which are red round. Now, does this help?
Well, yeah. Looks like 3/4 of the pollen would be purple long pollen, and 1/4 would be red round pollen. This ratio makes sense if the two genes were somehow linked together and passed on as one gene during gamete formation.
Well, yeah, but what about the other data? We can't just ignore it.
Yeah, you're right. We can't ignore this data over here. Let's focus on the behavior of these homologous chromosomes during meiosis. Each one will duplicate and produce chromatids. Often, the chromatids will intertwine, like this. And then sometimes, they'll break. And the chromosome material will be relocated on the other homologue. This is called crossing over.
So that's where we get the capital P, small l, and the small p, capital L chromosomes.
Yes.
Okay, all right, I get it now. So the purple round and the red long offspring would be the result of crossing over. The homologous chromosomes exchange genetic material.
That explains why our predictions didn't match Punnett's results. Crossing over took place, which changed our expected ratio.
Crossing over is one of the primary mechanisms to produce variation in gametes. The individual chromosomes have a combination of DNA that originates from both parents.
Psst. Did you figure out why your prediction was wrong?
Maybe we can help you solve the puzzle.
We're chromosomes.
I came from the mother, so I'm a maternal chromosome.
And I came from the father, so I'm a paternal chromosome. We both contain genes.
And as you already know, genes determine the characteristics of all organisms.
During interphase, we produce copies of ourselves. Then, during meiosis, we separate and enter different gametes. You know that chromosomes assort independently, but that's not necessarily true for genes. They often do things differently, and that's your first clue. For example, if I have the gene capital A and the gene capital B, and my homologue has the small a and small b genes, then the gametes will be capital A, capital B, capital A, small b, small a, capital B, and small a, small b gametes. Now here's another hint. What you see isn't always what you get. Chromosomes can trade genes.
It took them 8 years. Finally, Morgan had his answer, or rather 2 answers. He discovered that some genes on the same chromosome are linked together. During meiosis, when many characteristics separate independently, these linked genes stay together and are inherited as a unit. Morgan also discovered that during meiosis, some genes move from one chromosome to another by a process called crossing over. Let's go back to that sweet pea experiment for a moment. Remember, one homologous chromosome had the genes capital P and capital L. The other homologue had the genes small p and small l. It turns out that the genes for color and pollen length are linked together, so the resulting gametes were capital P capital L and small p small l. The offspring of that cross were 3/4 purple long pollen and 1/4 red round pollen. But we also discovered purple round and red long pollen. How do you account for them? They were produced by crossing over. During meiosis, intertwined chromatids exchanged genetic material producing 4 gametes: capital P capital L, capital P small l, small p capital L, small p small l. When the gametes combined with each other, we observed a change in our expected ratio.